Well, we're going to talk today about the systems biology of malaria. It's no surprise that the room is half empty whenever we say the word math. People say it burns, why do you do this to me? And I flunked in calculus, etc. It's not as complicated as it sounds, if it sounds complicated at all. So let's start with the two projects that have been mentioned today. One of those projects is the MAPIC project. Mary just gave us a wonderful description of this project. We expect this project to give us so much information about the disease. It's a unique data set. We had never looked at any disease with this level of detail. And this poses a series of problems. I'm going to comment on some of those problems because nobody has really done this before. So we have strategies in place, and we expect some of them will work. We expect we will discover new strategies to exploit the data. The other project, I'm a lucky baby professor because I'm a co-investigator in the two largest malaria projects right now, doing data management and mathematical modeling. The Isomer project is the International Centers of Excellence in Malaria Research, 10 groups around the world. Some of those groups are providing samples for MAPIC. And we look at human disease, epidemiological, we look at entomological data. At least 1/3 of each project, each one of these groups, is looking into mosquitoes. And as people mentioned before, they are very important for epidemiology. I'm going to show some very interesting data to emphasize that aspect. And these are the collaborators. Let me start with a quote I always like to show whenever I talk to mathematics, about mathematics to biologists. This is a quote from, let's say, if any of you know it, since I have shown it several times, this is a quiz. Maybe some of you remember. I simply wish in a matter which so closely concerns the well-being of mankind, no decision shall be made without all the knowledge which a little analysis and calculation can provide. This is just quantifying what we know. This is Daniel Bernoulli in 1766, a while ago. So this mathematical biology is not new, has been here from the beginning. It's here, so you better learn it. You're late in the game. The next quote is, this is also very true for the type of things that we do. Do not seek so much to understand the answers as much as to understand the question better. And this quote comes from Athens, Georgia. This is Hibachi Grill Supreme Buffet. That was a fortune cookie, and it's true. I kept it as a proof that it's true. Now, my first son was born on Sunday. So, of course, you know, every parent is very proud of their sons, daughters, and so this incredibly beautiful, handsome young man is the father of the baby. But let's not forget the following. When we talk about children, for me it would be devastating if I lost my child. But this happens every minute to malaria. Hundreds of thousands of children die every year of the disease. We got the perspective of somebody who had malaria. It's truly a devastating disease. No other disease comes close as a scourge for humanity. Excuse me. Unfortunately, by the time we finish, 30 children would have died of malaria. By the time I finish my talk. So this is an ongoing tragedy that we just have to stop. That's why the isomers and MAPIC are so important. We expect to be able to provide some insight into a possible solution. Now, of course, you have seen this gentleman, Sir Donald Ross, and I want to emphasize that he was an Indian-born British polymath, emphasis on math. He was a novel writer, poetry writer. He was a physician, a very controversial figure. He liked to fight with his colleagues. He got a very bad reputation. He got the Nobel Prize in a fight because the actual person who did the discovery was a different person. And he appears in every picture that you see except the Wikipedia picture, maybe a couple more. There is an exhibition, online exhibition, and you will see tons of pictures of Sir Donald Ross. always appears with a microscope. So you might be tempted to think he was a microscope salesman. But no, the microscope was the tool of the day. Now we are ahead of that point, and we have different tools, different microscopes. So let me just signal 4 points that are critical points, transitional points in the application of mathematics to biology. The first point is 1766. You saw the quote, so if you say it again, you remember. Daniel Bernoulli, 1766. Survival rates of smallpox via inoculation. So he made a small calculation. That's the small calculation he was talking about. An integral. Those of you who thought that calculus didn't work, didn't have a utility in real life, actually it does. Small integral, and then he was able to say, I'm going to save this many lives. A few years later, it was confirmed because the decrease in the number of cases was signaling that he was right. 1916, Sir Donald Ross. He made what he claimed to be his most important contribution, a mathematical model. A mathematical model known as the Ross model, and we will say it in the next slide. Then Kermack and McKendrick took what he had done, and they worked on a new family of models that are the standard way of doing quantitative epidemiology nowadays. Then in 1954, McDonald added the delay it occurs between infection in the mosquito and transmission to humans, the 2-week lapse. And these are the famous SEIR family of models. Extremely simple concept, extremely simple. You divide your population into compartments. So you have susceptible people, they go into the first compartment. You have exposed people, then you have another group of people. So you are either in one group or the other group. And we count how do we gain people in each compartment. So susceptibles, well, people get recovered from malaria and they become susceptible again. So you can imagine that there is a positive quantity that involves the number of people being recovered. How do we lose people from the susceptible compartment? Well, we lose them because they enter in contact either with a vector or an infected individual, and then they become exposed or infected individuals. Exposed is this latent state in which individuals are not yet infected, and so on and so forth. And there are many variations of this model. But here is the trick. Before I punish you with equations today, whenever you see the quantities that we're going to look at, this is what you have to think of. Either a velocimeter or an odometer. What is a velocimeter? The velocimeter simply measures the rate of change. If you're driving your car, it's the rate of change of the distance. You're covering a lot of distance in a short amount of time, or a longer distance in the same amount of time. That's the velocimeter. That is a derivative. So whatever I tell you, this is the derivative of, or wherever you see a variable with a dot on top of it, which is the standard notation, or d, quantity, over dt, all you have to think about is a velocimeter. That's all there is. And of course, if you are moving in your car, or you have a population of cells changing in the host system, you have the absolute numbers at every point in time, and that's the odometer, and then you have the rate of change. We are interested in the rate of change. If things change fast, well, it depends on the context. If you have a tumor and the tumor is changing fast, hopefully it's decreasing fast, because if it is growing, you're in trouble. If you have a tumor and the tumor is not growing, the rate of change is 0, you're in a state of equilibrium, nothing happens, nobody worries about it. Okay, so let me just comment quickly on the two types of models that were first introduced by Ross and McDonald. Ross said, remember, derivative, the velocimeter, how fast are we producing infected humans? I sub h means infected humans. Well, it's the product of 3 quantities times the infected mosquito. Is the product of infected mosquitoes and 1 minus infected humans. Well, infected humans really is the proportion of humans that are infected. So if you have 30% of the population being infected. 1 minus 30% is 70%. That's the population who is not infected. I'm sorry, it's the opposite. I just said the opposite. So if an infected mosquito bites a non-infected person, they might become infected. And they recover at a certain rate, and this is some expression for the mosquito. McDonald introduced some exquisite complications here, the delay. I'm not going to go into the detail, but what is important is that a quantity can be calculated from here. It's quantity known as R naught, or R sub 0. If this quantity is less than 1, it means that for every new case of infection, let's say you have 10 new cases of infection and your R naught is 0.9. That means that these 10 people will be able to infect 9 people. And then these 9 people will be able to infect, well, a little bit more than 8 people. So the infection is decreasing in the population. If this factor is greater than 1, then where is my pointer? Here. Greater than 1, the number of infected people is going to increase. So 10 people might infect 12. 12 might infect 14, and so on and so forth. It keeps growing. Okay, so now that's at the population level. Now let's see what happens inside the host. You have a network of interactions. And what we want to measure inside the host, it's a multi-scale situation. First, we're very interested in the population of each cell type. Why is that? Because if a process is taking place inside the host that involves a complicated chemical reaction, some metabolic pathway, but that process is not interfering with the state of the host, we really don't care about that reaction. We really don't care about the phenomenon that is taking place. Why? Because it's not a state of disease. We're interested in the states of disease. We're only interested in those processes, those networks that actually interfere with the cellular processes. So we start at the host process, analogous in an analogous situation to when we study populations, with the populations of all the types of cells that we have. And all the interactions they can have. Eventually, our goal is if we could understand all the reactions, then of course we wouldn't need to torture the monkeys and different animal models. We would have in silico models, and that would take us to the next stage. Given the fact that I don't have much time today, I'm going to go over this just like this. Forget about the equations. We don't have time to explain them. These equations, what matters is those numbers and things that flashed, are ways we have, this is the laboratory of the mathematician, are ways we have to calculate where the system is going. So if I have enough measures about what's going on in a population, that's the isomer project, or inside a host, that's the MAPIC project, I will be able to tell where the system is going next, the state of epidemiological equilibrium, or how is the individual going to survive the disease, or how is the individual going to die of the disease. This slide simply shows that these models, the classical models, can be used in many combinations. So for example, this particular model is by Prosper, 2012. That's her dissertation, University of Florida. And this lady was trying to see what happens when you have competition between two strains and which one is going to win over the long term. And turns out one of them does. And there is a nice mathematical formulation for that case that has very important implications. The other thing that we have to take into account is the scale. So if we start from molecules and go all the way to helminths, if we take this scale and keep going in the same length that we have here, we get to the continental scale. So when we study the malaria problem, we have a gigantic scale we have to study, going from molecules, those metabolites that we're measuring, the transcripts that we can measure, going to the cellular populations, and moving eventually to the epidemiological component. And we can do it. Actually, we're doing it. It's happening right now. MAPIC and the isomer projects will finish simultaneously by 2017. At that point, we hope that we will have enough data to produce a comprehensive model that goes from molecules to the continental scale. But we have a problem, and it's that the current models do not work for several reasons. One of those reasons, or probably the most easy to understand for a biological audience, are this. You have microdiversity, you have simultaneous infection, you have antigenic diversity, and you have antigenic variation. There are places in which malaria is prevalent, in which 90% of the population is infected, but many of them are asymptomatic. And that's a gigantic problem. If we go and measure on the field what's going on, we will see a bunch of symptomatic people that might be seeking active clinical support. And then we see a gigantic population that is asymptomatic. What is coming out from the field is that in some places, we have 10 times more asymptomatic cases as compared to symptomatic cases. Now, keep that in mind because that's extremely problematic. The other thing that we have is vector behavior. This is fresh data as well from 6 months ago. This is 6 p.m. This is the percentage of people who are awake and up and walking around. By midnight, everybody's in bed, and we assume that they are protected in this white area. So if they are in bed, they are under a bed net, and they're protected. But you see that mosquitoes start their activity before people go to bed. So even if you're using bed nets, you're still exposed. And we can measure the loss of effectivity of the bed net due to mosquito behavior, another math model for this. It's a nice integral. Now observe this. This is one location, and you have the peak of the mosquito here. You have another location, and the peak of the mosquito happens with everybody is up. So you would expect totally different epidemiological scenarios, right? So which one do you think is worse, this or this? Make a guess to see if you're awake. Quiz. The second one? Okay, so remember the name. This is Nariño, the other is Córdoba. Remember those names. Because we're going to see, this is Nariño and this is Córdoba. Oh, the opposite of what you thought. When you look at clinical cases, what gets reported to the public health systems and the World Health Organization. But if you look at asymptomatic cases, we have the totally opposite situation. So this is an extremely interesting slide. Let me just point a few facts here. Vivax. You have a lot of vivax in this region. You have very few vivax in this region. The amount of vivax decreases going south. The amount of asymptomatics increases going south. But the proportion of vivax to falciparum is inverse with respect to the symptomatic cases. There is a very interesting population dynamics happening here. So we know that the population of the parasite is very heterogeneous. And that's one of the problems that we have here with antigenic diversity and antigenic variation. Unfortunately, we don't have enough time to go into the modeling details of how do we incorporate this into a population, but think about the following. If you have 50 VAR genes and 6 loci, you really have a collection of VAR repertoires, collections that are expressed in populations. And as I mentioned, this lady Prosper from the University of Florida did an analysis of all the competing populations. Well, you might have families or strains that are expressing at a given time antigenic repertoires. And one of the hypotheses that is being thrown to the field in the mathematical modeling field for malaria right now is that you never lose your resistance to malaria. You never do. You get infected, you might be resistant to that specific strain and antigenic variation family at that point. What happens is it changes, and there are dominant types that keep changing over time. So what might happen is that since the population, the dominant population in the field is changing, and we have such a gigantic number of possibilities, the combinatoric possibilities so big. In 1 year, you might go to the field, and even though you're already immune to the previous year, you're not immune to the new year because of all the changes that have happened in the repertoire. Okay, let's see here, before I go into the math, because I know that I'm going to run out of time. How much time do I have?
You have 7 minutes.
7 minutes, okay. So I will be able to punish you. Good. I don't want to let you go without throwing a few equations. So we'll get there. But before we get there, I want to emphasize the following. In this slide, we have asymptomatic versus symptomatic cases. The problem is, in this moment, all the models, mathematical models that are available in the literature, and even those that are active modeling initiatives, Malaria map, for example, they rely on data that is being reported by national systems of health. There is very few, preciously few data about asymptomaticity around the world. But as we saw, that's where the big problem is. So in this case, in Colombia, all the efforts of the Ministry of Health are located in this area, Cordoba, when it's in the south where we have the biggest asymptomatic problem. So unless the asymptomatic cases are taken care of, it's impossible to eradicate malaria. And that's one of the big questions that are lingering right now in the community. Can we eradicate malaria? Well, it depends. What do you mean by that? Eradicate symptomatics or eradicate asymptomatics? Now, how do you go after the asymptomatics? That's why those tools that Dr. Kumari is developing are so extremely important. How do we get a population count of those who are asymptomatic? So we can fit this population correctly into our models, so we can predict what is the amount of public intervention that is needed to actually decrease that R naught number that I was commenting on before. If we can produce an R naught less than 1, eventually the infection will disappear from the population. And that would be the goal. But we just do not have enough information to do that yet. Now, what we want to produce is a map like this. This is a risk map, extremely different from the risk maps that are available in the literature right now. Because what we did is that. P represents the amount of pathogen, in this case Plasmodium, inside an individual. P sub I, the I means human. K, the subindex K, represents the strain. That's the genotype. The subindex 1 represents the first life stage of the parasite, second life stage of the parasite, third, fourth, etc., the gametocytes, and PJK, notice that it changed from I to J. J is the mosquito, the amount of pathogen in the mosquito. And then we do this, let's focus on these two equations. The first equation and the last equation, it has a function for the position of the humans and a function for the position of the mosquito. So if we can model every human in the planet and every mosquito in the planet, we might be able to predict what's happening with malaria. We don't need to do that. There are ways in which we can calculate the asymptotics of this system without actually running a simulation with the 1 billion mosquitoes. That would be extremely computational intensive and very non-informative, because we have methods to do this very quickly just with a little bit of math analysis. Well, let me go back before we close that. Any question about that? No, okay. The model that I was showing you is simply, it's based on a toy model. And of course, we start with toy models, as just go to a wet lab and start with very simple tools, and you keep building complexity until you can make something very sophisticated. We started with a toy model in math, in which we have a cell that it's a parasite, and one type of immune cell that is going to attack that parasite. It could be humoral immunity, dendritic cells, doesn't really matter. For the toy model, in the live model, of course it matters. But what it matters here is that the parasite population goes up. As a result of this increase in the parasite population, the immune response goes up, and this brings the parasite population down, and finally the body is able to get rid of the disease. That's the foundational concept of what we're trying to do, linking what we're learning in the MAPIC project, that is the within-host dynamics, and that's what this gigantic model eventually means, that we have the populations of the cells, of the different cell types in the parasite, in which all these coefficients that you see here, omega, tau, etc., phi, are calculated based on molecular information. And that's how we link populations to molecules. And since we have this for every human and every mosquito, that's how we link this information to the epidemiological scale. So we go from the molecule to the continent. Of course, this is a success, but the success will be visible in 2018 probably, 2017 when the two projects finish, and we have been able to analyze all the data. So I just leave you with a thank you. Thank you for your attention. So if you have any comments, disagree. Oh, you disagree. Go ahead.
Well, in terms of your simple model there with the parasite and the host cell, so the model that you're developing with the host cell, does that take into account, say, immune repertoire, looking at the, say, a T cell receptor or the B cell receptor, and what that allows the cell to react with against the different epitopes?
That's the goal. In this moment, we are not there yet because the MAPIC project has not even produced the first complete data set from the many experiments that we have, but we want to get there. Specifically, how are we going to do it? Well, we know the biology behind it. We have ideas about how to go to solve this problem. But really it's a question of, once we look at the data, what is the data telling us? So we really have to tailor whatever quantitative model we use to the sort of data that we have. We don't want to build a model that requires data that we cannot collect. We really have to go to the data that has been collected. So once we have all the data sets, I think that we will be able to do exactly that. And if we cannot do it for whatever reason and we think that's important, those are the iterations that Mary was talking about before. We might be able to refine that and request help from our biology friends. Ladies first. Oh, I'm sorry, go ahead. Oh, you do it.
No, all right. You're the speaker.
You're the boss. Ladies first. Okay.
So I wasn't able to grasp all of the aspects of the model when it was up on the slide.
No, really?
So I may be asking about something that was up there, but you were modeling different strains, clearly, many different strains. And I'm wondering so I'm assuming your model can address people who are infected with multiple strains, but if you do model that, do you assume that the strains act independently of each other?
No, not necessarily, because if you take a look at this wonderful model that we have here, we have a human eye, subindex K. So K can vary for every human eye, and K is the genotype.
Yeah, but if you have two strains in an individual human, are you assuming that those two strains act the same in that human as they would if they were by themselves?
Well, the way it works right now, and by no means is the perfect answer, but the way it has been implemented is that two different strains might interact here. If you look at the gametocytes, once they go into the mosquito, you might have diversity through sexual reproduction. And that's what we're considering at this stage.
Okay.
Different sorts of interaction. If there is evidence that we should be looking into that, of course, can be incorporated. But I just want to make a comment. I know that many of you feel intimidated by these equations, and you shouldn't, because it's extremely simple. Look, we have positive quantities. Positive quantities means something is making this particular population grow. That's all it means. We have negative quantities. Negative quantities means something is making this population decrease. So how does it decrease? Well, you might have something like this. You should be familiar with that Michaelis-Menten type of thing, saturation reaction. And there are many ways in which we can capture that. So really it's not that complicated. We just look at the data statistically. From the statistics, we try to determine what is the functional correlation between the variables, and that's what we use to build these models for rate of change. So,
Can you just add a comment
Go ahead.
to this question? In this population in Colombia, there are, as you may already know, there are very limited number of strains of the genotypes.
mhm.
We are also have some experience with this population. And clearly, I think Mary is probably referring to something like in Africa where
Totally
there are
different.
multiple strains. But in this population, especially in the two sites you mentioned in the down south, there are probably about 3 different clonal population in that area. There is well, we have the data on that. You know, related to HRP2 work we did, we had a chance to look at that population. We can talk later maybe.
I definitely want to talk to you because apparently we have many more than that, but definitely that's something that we should look at. And one of the important aspects of this sort of quantification, wait, where is my map? My map is here. Of this sort of quantification is the ability to estimate what is the diversity of parasites that we have. So in this map that I didn't tell you what it was, the red color in this case shows what is the higher intensity of, what is the environmental density of the parasite, which is totally different to building a map of clinical cases, as you notice, because of the symptomaticity. A map of clinical cases would have the red spots not here, but here, in a totally different location. And according to this map is different. So definitely parasite diversity is extremely important. And well, I don't know, we have to talk.
There's a queue of 2. Hassan, come in here.
So in that row of equations, I did not see a term for the actual geography of the plate.
Oh,
So, or is there one?
it is there. It is there. You're an acute observer. But let me tell you what it is. There it is. It's right here. It's right here. When you see the sum of some coefficient, which is J sub I, that is the rate of transmission from mosquito J to human I, times a function H. And this function H is a function of the distance between the human and the mosquito. That's where the geography is. And we have developed with another mathematician in the math department what we believe is a very elegant way to solve the geography problem. That's something that we definitely should talk about in the future here in UGA. And if you're interested in the geographic component, that's one of the most important directions that we're pursuing, because we realize the tremendous importance of human movement and natural barriers.
Well, I was also thinking in terms of, can it be predicted to tell you what's the right combination of geography, people, and mosquitoes
Yes.
that produces resistance?
Well, I don't know if it produces resistance, but I can tell you that with the isomer projects in this part in Colombia, we have only for Colombia like 20,000 mosquitoes. Each one of them was analyzed to see if they were infected with malaria. And we have the location where they were found. And we did the correlation. Well, let's see it. The simplest problem of them all, or the simplest approach. Linear regression between population density, temperature, precipitation, altitude, and these clusters come out beautifully. So we can tell in what parts of the country we expect what species of mosquito, and as we saw in these other graphics, each species in different locations have totally different behavior. Not only that, mosquitoes are the most fascinating creatures. I mean, it's probably second to Plasmodium in my list of favorite things. Absolutely fascinating what they do. If they put barriers and mosquitoes cannot fly, they start walking. This has been reported. They enter walking into the houses. And this has been reported in Peru and in Colombia. They change behavior. If you spray, let's say, curtains and walls, then they go under the furniture. And they stay there. So they're not as dumb as we assume they are in these mathematical models.
All right, now last question.
So yeah, I just wanted to ask about the asymptomatic population. It seems to me that they will never show up at the hospitals, and they will never be treated.
Correct.
And so, considering the fact that in the United States, throughout Louisiana up to Florida, that malaria was controlled in 1948 through 1950. My question is, using these models, why wouldn't we utilize the same approach used in the southeastern United States to calculate the rate of transition from asymptomatic to symptomatic infections, and then perhaps, based on that, do a mass treatment of the people.
Yeah.
What do you think about that?
Well, that's a very interesting thought, and I think that some approaches historically have been intended in that direction. I think that if we could implement in all these countries the same phenomenon that occurred in the US, that limited the exposure of people to the mosquito, that would be a tremendous success. That is improvement of economic conditions, air conditioning for everybody, better sanitation, and a massive public health intervention to kill mosquitoes during a prolonged period of time that was longer than election cycles. That might not be possible in other parts of the planet. It's extremely difficult. Mass treatment has been attempted in Brazil, in Guyane, when they were mixing, biologists please fill the blank, eggs with salt, and that produced very limited results. So mass treatment, it's complicated because not all the malarial drugs are, remember what the other say was?
Yeah.
They're not said. So I don't know, that's a problem that it's open, and I think the best way we could attack this problem is if we understood what is the size of the problem that we have and how is it changing, what is the derivative of this problem, because we just don't know. Hopefully ISOMER will give us all that data.
I've been hearing that air conditioning is one of those big phenomena, but in 1940, not everybody had air conditioning, and there were lots of very poor populations, mostly in the south, and people who lived and didn't have shoes. So you managed to maybe drop down that level of transmission, but I think that's a very interesting situation because you didn't eliminate everything, and not everybody got completely covered and protected.
Exactly.
So that's a very interesting concept.
It is.
But
And
you remove the vector.
yes.
That's big.
And not only that, the vectors are still here. Yeah, you remove them temporarily, but what happened is we removed, for a period of time, the population of the Plasmodium. Our goal is not to kill mosquitoes. They are fascinating creatures. They're not mean. I mean, they're obnoxious. But that's it. We shouldn't kill mosquitoes. We have to kill the Plasmodium. That's what we have to do. So for that particular thing that you just mentioned, here it is the solution. This little term, which is P minus sigma, where P is the population of the Plasmodium, has a very particular name. It's called the Ali effect. The Ali effect takes the name from a book from 1938, a biologist from Chicago, who wrote a book that was quite popular, The Social Life of Animals, and he made the observation, or he documented the observation, that when you have populations with very low numbers, the population might go extinct. And that has been observed in many situations, ranging from plankton to small mammals, big mammals, etc. So that might have played a role, because the population of the Plasmodium was so small, and perhaps there were pockets of Plasmodium, but there were not at the same time pockets of mosquitoes, mosquito populations. They just died. And that's what we would like to achieve in any case.
All right, Juan, I'd like to thank you for sharing your mathematical view of the problem.
Okay, thank you. Oh, yes.